If you take all of the diversity of animals today and trace their evolution backwards, right to the very beginning, which was the first group to evolve? We used to know. Now we kind of don't. Animal evolution is complicated. There's an awful lot of it. So there are something like 30 phyla. These are the major divisions of animals, of which one, for example, is chordata, the chordates. And this includes everything from the simplest fish up to really complex vertebrates like, for example, toads. Humans fit in there somewhere as well. So that's one out of about 30 phyla. The chordates fall into the wider group called deuterostomes, which is defined on embryology, and this we share with things like starfish, obviously. So this is a fairly fundamental group, and the basis of it is the embryology, how we actually develop in the early stages. The counterpoint to that is the protistomes, which are now divided into two major groups, the Lophotrochozoa and the Ecdysozoa, depending on whether they molt, and some complex characters for the Lophotrochozoa. And most of the phyla of animals that you'll be familiar with, so annelid worms, mollusks, brachiopods maybe, all of these things fit into the Lophotrochozoa and the Ecdysozoa. All of those have fundamental bilateral symmetry, which puts them into this extremely expansive group that includes most animals called the bilateria. Even starfish have bilateral symmetry, they're just sort of pretending to have pentaradial. Even we have bilateral technically, although, you know, heart's a bit displaced off to one side, and we're not perfectly bilateral, but that's good enough. So this fundamentally bilaterally symmetric group is what is most of animals, and what is considered, in quotes, higher animals. Because there are also the basal groups, and this is where the huge controversy is today. There are four groups of animals that do not have bilateral symmetry and are considered, based on all sorts of evidence, some genetics and morphology, to have evolved and separated from the rest of animals before any of the bilaterally symmetric ones evolved. Some of these are familiar, some less so. Some we won't deal with much at all because there's not much we can say about them. So I'll get the placozoans out of the way first. These are tiny, apparently very reduced, totally amorphous. Think of something like a flatworm with no internal organs and no shape and nothing really about it at all. It's a little blob with a wiggly outline that moves around and absorbs food directly through its surfaces. It's an incredibly interesting animal. It's just that we know so little about its ancestry because it's off there on its own, and it's probably secondarily simplified. It probably came from something a little more complex and has lost most of its features in this quest to be as, well, adaptable as possible by not having anything as part of it. Then there are the cnidarians. This is the most familiar one to most people because it includes jellyfish and sea anemones, things that are familiar from the seaside. And this is quite a large group. They mostly are based around tetraradial symmetry. So they have sort of radial symmetry, circular, but with normally sort of fourfold division. That's the fundamental basis, but there's variations around that as well. They're also all defined by having stinging cells, the nidocysts, which are basically tiny little harpoons that can be fired out from the tentacles and can be quite painful or deadly in some cases. Next one is the ctenophores. These are a truly magical group of weird and wonderful critters that are not so familiar. They are known as sea gooseberries, some of them. They do turn up along coastlines, but comb jellies is the popular name for them. And these are really quite complex animals. They have rows of, well, what they call comb rows, but little lines of cilia that around down the side that beat and draw water currents around. They have a statolith, which is a sort of balance organ. Many of them have tentacles. They're active predators. They secrete a sort of sticky substance to catch their prey with rather than the stinging cells. And they're wonderful things that everyone should get to know better. And the fourth group, which has traditionally been the simplest of the lot, are the sponges. And these are effectively filter feeding. They are basically blobs that sit on the seafloor, are extremely porous. They suck water in through holes, tiny, tiny pores, and then spit the water out after having got all the nutrients out of it that they can. Unlike the ctenophores, which have a sort of eightfold symmetry, the sponges today are either radially symmetric, just with a circular symmetry, or they're asymmetric, amorphous. Those are your four major groups of animals at the base of their evolution. And the question is, which came first? Which one is most like the ancestral animal, and therefore which represents the base of all animal evolution? Which one shows us where we all came from? Before we get into that, though, we first have to define what exactly we mean by that question. Because there is sometimes an assumption that we have these phyla today, the cnidarians or the ctenophores, and you follow them back and they are cnidarians or ctenophores until suddenly you get to a point where they're not. And that if you have anything on that branch leading to modern ctenophores, it will be a ctenophore. But it doesn't necessarily have to be recognisably a ctenophore. What we're trying to do at the moment from genetics, or what geneticists are trying to do, because it's not me, is work out the topology of it, by which I mean the arrangement of the branches in the tree. Which branches came first, irrespective of what the animals on the early parts of those branches actually look like. So what we're looking for here is the earliest branching group, the one that has split off first, so that all of the other groups of animals are more closely related to each other than they are to this one, which therefore theoretically better represents the ancestral condition of animals. And this, of course, is theoretical because you can have simplification. Just look at sea squirts. You've got to love something that swims around as a tadpole, attaches itself to a rock by its mouth, and then resorbs its brain because it doesn't need it anymore. So that sort of thing is possible. What we're looking at here is the first stage of it. We're trying to pin down the actual arrangement of branches, which is of course related to our ideas of how animals evolved. But the fundamental point to start with is which ones branched first. If you look in most textbooks, or if you even look in papers or ask an evolutionary biologist, the answer you'll get is probably fairly straightforward, that the answer is sponges. This goes back a long way in that we have had ideas that sponges are basically one step up from being colonial protists. There is a closely related group of single-celled organisms. They are closely related to animals, they're our next nearest relatives, called choanoflagellates. And these things are single cells that very closely resemble the cells in sponges that do the filter feeding and actually create the currents. So there has been an idea that you basically have a film of these things living colonially, which then becomes slightly more complex, secretes a few more cells, these cells cooperate with each other, and you end up with a network that filters water and does it very effectively, and that is the origin of animals. That is the long-standing idea that has been argued for many, many years and has been assumed for many years. And you'll find this, as I say, still in textbooks all over the place. The problem is that when you look at the detailed evidence for it, it starts to break down. And especially now in the age of genetics, when we're interrogating the earliest reaches of animal evolution, it no longer seems to make sense. It may still be that sponges are the first branch, but it also may not be. And the difficulties are such in resolving this that it looks like the last common ancestor of sponges and other animals was probably significantly more complex than a colonial protist. For those who don't understand how we do this with genetics, yeah, you're not alone. But it's quite a simple idea in principle. We have the ability now to sequence the genetics of many organisms very, very quickly. And so there is now a database of the genetic codes for particular genes, for different parts of the genome, and so on, for a large range of different animals and other things. And what you can do is then make a huge great data matrix where you identify the same genes or the same sections of the genome for 100 different species, put them into a supercomputer to analyze, and it basically looks at every possible arrangement of those animals into a tree and gives you the most likely arrangement based on the similarities of the genetics. I have to be careful saying most likely because there are all sorts of other variations on that as well. It's an extremely technical area. There are many subtly different approaches and they can give you quite different answers. But the process is basically that. And it should be really quite accurate, and it is, especially for the younger groups of animals. So when you're looking at branches that have diverged relatively recently, then we can normally reconstruct their evolution, or the sequence of evolution, relatively well and reliably, and we don't get too much conflict. When you go back to the earliest animals, though, it's a long, long time ago, and a lot has happened to the genes in that time. Not only have things just evolved, you also get things like lateral gene transfer, where viruses can move bits of genetics from one lineage to another entirely. You can get all sorts of other peculiar things going on. Sorry, just distracted by a ladybird. Oh well. And that can really get in the way of a clear signal. The further back you go, and the more changed the organisms are that you are trying to look at the ancestry of, the harder it gets. And this is where you can get unexpected results turning up. The trouble really started with a paper in 2008 and then more in 2014, and then it started accelerating, where genetic results looking at the genes of various early branching animal groups, such as the cnidarians and the ctenophores and sponges, started to find topologies, arrangements of the branches that did not match. Specifically, they found topologies with ctenophores coming out as the oldest branching animals. Yes, ctenophores, those complex things. This led to a large number of papers over the last 10 years where people have made more and more detailed analyses, meta-analyses, trying to tease apart the lines of evidence, the weak points in these arguments, trying to work out how to actually resolve which of the conflicting sets of results was correct. And that debate, in short, is still ongoing. To try to get to the bottom of this, it's no longer even just about the genetics. It's going into all elements of the molecular biology, the biochemistry of these things, trying to find lines of evidence supporting one side or another. So nitric oxide signalling, for example, supports the ctenophores first. If you look at the number of transcription factors within the genetics, then you end up with all the simpler things have fewer. But that doesn't necessarily translate to them being primitive in the sense of being early branching. There was a paper on gene linkages, also suggesting that ctenophores branched first. But then there's another paper after that which is starting to cast doubt on the exact methodology used to generate this and biases within the analytical techniques. It's not even as if these are the only two options, because there have been some results suggesting that placozoans came first. These are probably errors. I think the consensus is that they're slightly higher up the tree. But then there's yet another version, which is not ctenophores or sponges, but ctenophores and sponges. This is a scenario called the paranomalia, which doesn't turn up very often, or at least isn't reported very often. But there was one paper that showed that with very slight changes in your analytical approaches, that's the answer that you can get, and apparently quite convincingly. Personally, I think that's a really interesting idea to approach, because from a paleontological perspective, there are some striking similarities between some fossils interpreted as very early sponges and others interpreted as very early ctenophores. And there's a long way to go on that debate yet, but we'll come back to that in another video, I think. At the moment, it's safe to say that this is not a popular perspective. Of course, it's not just the analyses themselves, because all of these scenarios have implications, and you have to explore the results of those implications, whether they imply things that are even possible. So, sponges and ctenophores in different ways lack various features that might be shared between other groups of animals. So if the ctenophores come first, does that mean the sponges have lost them? Things like a nervous system I'm talking about here. Or does it mean that the nervous system of ctenophores was evolved independently of that in animals? And it looks like they have developed them independently, but they have certain similarities at the roots of the structures that were then developed. Another one is epithelia, and we discussed this a little bit in the talk about chancelloriates. So this was dealt with by Sachkova 2023. Let me just read you a little bit. For example, epithelia had been considered conserved throughout animals and key for the evolution of metazoans. However, recent studies changed this notion by revealing that ctenophores that are equipped with an epithelium are lacking some proteins key for the organisation of epithelium in bilaterians, while sponges have a more complex epithelial toolkit but mostly lack a true epithelium. This is really interesting because on the face of it, that looks like sponges have everything they need to create a true sort of epithelium, this dermal layer, a discrete sort of outer layer of the cell tissues. But for some reason they don't do it. Is that because they never evolved it, or is it because they've lost it, because they've evolved down a different route? And it's all this exploration that is going on to try to work out what is possible, what is reasonable, what isn't. That is also tied in inextricably with the analyses themselves that are generating these different scenarios. Because we used to have a really nice, simple explanation of protist, colonial, sponge, ta-da! But it doesn't work that way anymore, it seems. The more we look into it, the more complex the whole situation gets. It's got to the point where it's these tiny little bits of apparently abstruse information to most people that are turning out to be critical, because we have to really critically analyse every little bit of evidence in order to build up enough of an argument to go in one direction or the other. So how do we resolve it? It would be nice to think that at some point we'll get a clear answer from the genetics, from the molecular biology, but it's not necessarily looking that way at the moment. It's just too complex and too long ago to be able to get a clear diagnosis. There are so many bits of conflicting evidence that probably we're going to need something extra. It seems increasingly likely that these branches all happened in such quick succession that there wasn't much difference between them. So there can't be a clear answer because they were all very close in those early days. And of course we do have something extra because we have the fossil record. The problem is the fossil record is also extremely contentious when you go back to these first animals. I'll be doing quite a few more videos on this sort of thing. But in the Cambrian, we mostly know what things are nowadays. In the Precambrian, where the origins of the animals would have been, we have a wide array of fossils. We have extraordinarily strange and weird and wonderful things, some of which have been called cnidarians, and I think probably convincingly. Some have been called sponges, much less convincingly. Some have been called ctenophores, but I'm not sure many people really accept that one. But this is something I'm going to have to dive into in other videos to try to see what exactly we can tease out of the fossil record. But it does seem that the Ediacarans, this biota from immediately before the Cambrian explosion, when we get truly recognisable animals in all their diversity, these Ediacarans must be critical to understanding the origin of animals in some way, but maybe not in a direct way. And we also need to approach this from the other direction, from tracing these modern lineages back through time to their origins in the Cambrian, where we can recognise them, and look at the trend, see if we can extrapolate back to what they evolved from. And we're doing this now. There's a lot of work recently on ctenophores, which today are mostly sort of free-swimming things with tentacles and 8 comb rows, showing that they were much more diverse in their early history. There's still a little bit of debate, I suspect, over whether all of the things that are now assigned to early ctenophores really were ctenophores. But some of them have many more comb rows, and some of them have a sort of sclerotized skeleton. Some of them appear to have been benthic, so living on the seafloor rather than free swimming. They look like quite different animals. Sponges also, again, I'll do lots of videos on this sort of stuff, but they today, most of them are amorphous, blobby, encrusting things with no symmetry. But that seems to be a derived trait, that you follow them back in time and they become solitary, vase-shaped, a single osculum at the top, and that is their ancestral condition. As far as we can make out from every class, you trace them back, they converge on something vase-shaped with a hole at the top, which is not dissimilar to ctenophores. So one thing we can say from the fossil record is that it really doesn't support anymore at all this idea that the first sponges were amorphous and blobby and one step up from a biofilm, the protists. And therefore, when you are looking, trying to understand what the first animal looked like, or at least the first animal that was the direct ancestor of all of the animals alive today, then we have to take these things into account, because for all we know, there could have been a lot of evolution between that first ancestor and the first fossils that we recognise as belonging to those groups. So the first ctenophore, the first sponge, the first cnidarian, they may all have diverged a lot from something that was ancestral to all of them. And that's the real challenge of interpreting very, very difficult fossils, like the Ediacaran biota. There is a lot of speculation, I think, nowadays, at least informally, that you can go to look at the Ediacaran biota classic things like Chania, these weird quilted frond-like creatures, and fit those in because there's so many unknowns. But maybe something like that was the first ancestor of all animals. Personally, I don't buy that, because it has a different organisation of the body entirely. So it's possible that that evolved from something which was also much simpler and then evolved into the other groups as well. But it's very, very difficult, at least for me, to see how you can go from something which appears to be a sort of quilted frond that fed by absorbing nutrients directly from the water, to something which has the symmetry structures, the body form, the architecture of an cnidarian, a ctenophore, or an early sponge. But this is where the debates lie. And this is where we will have great fun exploring as I do more of these videos, and hopefully we'll try to get to at least the questions that are critical, because there is yet no answer. I can't just give you a new story that solves it all, because none of them do. But it is great fun trying to find out, isn't it? Okay, I shall leave it there. But the take-home message then is that we don't yet know. Was it sponges first? Was it ctenophores first? Was it sponges plus ctenophores first? Not sure. The evidence is still accumulating on both sides, the arguments and counter-arguments are still flowing, but one thing that is becoming clear is that that last common ancestor of all living animals was more complex than we used to think. That it certainly wasn't just a sponge, and it may have had features much more like a polyp or a comb jelly or something of that nature. But time will tell, and hopefully you'll be back to see what happens with me. All right then, I shall see you again.